Hey everyone, it's your Professor Christina Howard, and this video is going to be about the histology of the reproductive system, since the one I recorded on Zoom was so gosh darn laggy and interrupted a bunch. By just the slowness of my computer, I decided to make a completely separate video for it. So with that, away we go. So I'm going to go down to reproductive system, which as you can see is includes female and male. So we're going to just go through and do male first. And that's because there are a couple more structures than for female to learn. So the first thing you need to learn is identifying the testis versus the epididymis. So to do that, I'm going to pick a slide with both on there. So if you look closely, you'll see that under epididymis and under testis, there's this slide that is both. I'm going to pick that one. One thing you don't want to do is pick one that says neonatal and adult testis, because the neonatal testis is the one of a newborn, and that's obviously an individual that has yet to undergo puberty, which means that their testicular tissue is going to look way, way different than a sexually mature adult. So we're going to focus on people that can actually make spermatozoa, not people who can't yet. So I'm going to first go here and grab my Epic pen. Excellent. Okay, so I find that yellow shows up best for most things. So before I even zoom in on anything, what I'm going to do is just outline. This tissue is the testis. And this is the epididymis. So the epididymis is a comma-shaped organ that sits on top of each testis, which means that it's going to be smaller in comparison to the testicular tissue and separated from it by a bit of connective tissue, which you can actually see here between the two yellow outlined areas. So I'm going to start by zooming in on the testicular tissue itself, but first I do want to point out this area right here, which is between the two, and this is the tunica albuginea. Which means white tunic, but it's a dense connective tissue covering that separates the testis from the epididymis. This term will come back when we start to talk about penis histology, so keep track of that term. So to give you some context for what the testis is actually made of, it's a bunch of tubes wadded up, and the development of spermatozoa happens inside of those tubes. So in order to see cells like the Sertoli cells and the cells of spermatogenesis, including spermatogonia, spermatocytes, and so forth, we need to zoom way, way into the testis area. And I'm going to find one of these tubes that I think has the best set of examples of those cells. Come on. If I can zoom in, that is. There. Apparently I can only do it by pinching. That's fine. So what I'm doing now is I'm cruising around the slide and I'm trying to find examples of seminiferous tubule, which is, lots and lots of these are, that are more or less representative and have also a lot of diversity of cells for us to look at. So I'm seeing a couple that I like. And this is another sort of friendly reminder that starting at a low magnification and then noting what you see as you zoom in is the smarter way to work through this histology stuff, because you have limited time to learn these things and we want to work smarter, not harder, right? So I've found a seminiferous tubule that I find appropriate for teaching from. So let me outline it first. So I'm going to go ahead and outline it in yellow. And that is one. So this is just one cross-section of a convoluted tube that is making up one of the lobules of the testis. So if you look over here, what you'll see is each lobule contains 1 to 4 tubes, but those tubes are coiled up. And what makes a lobule? Well, it's a group of tubes separated from adjacent groups of tubes by connective tissue. So that's what that is. So now we're going to focus on the spermatogenesis cells and the Sertoli cells. So the deal with Sertoli cells, which are also called sustentacular cells, and I'll spell that for you right here. So these are cells that support and aid the development of sperm. So you can think of them as being nurse cells, and sometimes they're actually called nurse cells as well. So Sertoli is the name that acknowledges the person that discovered them. Sustentacular is a name that describes their function scientifically, which is to help nourish and sustain spermatogenesis, and then nurse cells is the shorthand version of that. Any of those are acceptable on a lab test. Okay, so really all you can see of these Sertoli cells, because they're large and columnar, but they're obscured by the developing spermatocytes, all you can really see are their nuclei. So their nuclei, and I'm going to make my dot smaller just so I don't accidentally cover over what I'm trying to circle. Their nuclei have this characteristic appearance where they are lighter staining and have a darker colored nucleolus. So what I'm doing now are circling a bunch of Sertoli cell nuclei. So remember, and I'm going to write this explicitly as well, this is the nucleus of a Sertoli cell. So if I were to draw the rest of the cell, which you can't really see the borders of here, what you would see would look something like this. And then maybe there's a neighboring one over here. We can't see the nucleus of it, but it looks like that. And this. And that. So those are the nuclei of Sertoli cells, and then I've drawn the imagined outlines. So let's write that down too. Can't see outline, but looks like this. So what I would encourage you to do is go look at the drawing of spermatogenesis in your textbook and compare it with what you're seeing on the slides to help you get a better idea of the morphology of the Sertoli cells. So that's the first cell type, and I'm going to switch colors and also undo my outlines because I want to use other cells. Oh no! There we go. It's a nice thing about the undo button. You can redo as well. So I'm getting rid of this just because I want extra room to be able to point out other cells. So I'm going to switch to an orange color. And now we're going to go hunting for spermatogonia. So spermatogonia are stem cells, and these stem cells undergo mitosis and give rise to another spermatogonium and a cell that becomes a spermatocyte. So these spermatogonia stay against the walls. Maybe blue would stand out more. Oh yeah, that's better. I'm going to change to blue. So the spermatogonia stay against the walls of the seminiferous tubule, and then when they divide, one of the daughter cells stays at the wall and remains a spermatogonium. The other one becomes a primary spermatocyte. So typical of the spermatogonium cells, they have a round, very dark nucleus, and they're always right up against the basement membrane. So I'm going to make an arrow for these two. These are spermatogonia. There are primary and secondary spermatocytes, but I'm not going to ask you to differentiate between the two of them because it can be really tricky. So instead, what we're going to do is use another color, this green color, to circle a couple of those. So they're just toward the lumen from the spermatogonia. They tend to have a dark nucleus. Not this one. This is another Sertoli cell nucleus. Ooh, here's a really good one. There's another one. So those are going to be primary spermatocytes. So your major hint is the nucleus looks a little bit diffuse compared to the spermatogonia, and they are away from the wall. Okay, so spermatids. Spermatids actually come in two varieties. So one of the varieties is, I think pink is going to be too, I'll try white. Pink is going to be too similar to the tissue color, I'm afraid. Well, let's try it. So spermatids are either these small, oh no, that's just fine, small little dots, because after the mitotic division, I'm using mitosis with a T, spermatogonia undergo mitosis, the spermatocyte starts to undergo meiosis. So it's going to divide its DNA and itself twice to produce cells with a haploid genome and cells which are smaller. So smaller nucleus, smaller cell. So what you get are these little bitty round guys. Oops, not that one. Little bitty round guys here. And you can see they're all over the place. They're pretty easy to find. So those are spermatids, one kind of them. Looks like I need to make a little white space for myself to draw in. So those are one kind of spermatid. The other kind of spermatid, which you can't really see here, are ones which look like these pointy spermatozoa in the middle, but which are embedded in the edge of the seminiferous tubule. So there aren't any in this particular seminiferous tubule. Excuse me. But you can see these. So the difference between a spermatid that is actually sperm-shaped and one that is round, like the one I'm showing you, is a process called spermiation. So spermiation is the conversion of round but genetically complete gametes into genetically complete gametes that can also swim. So spermiation is when they grow their head, they grow their midpiece, they grow their tail, and they become what's called motile, which means capable of swimming. So that leaves me with the final thing in here, which are these spermatozoa in the middle here. So we only call them spermatozoa once they've fully released from the Sertoli cells. You get the idea. So those are spermatozoa. Okay, so now I'm going to take a screenshot of this and we will move on to the next topic. Alrighty, so let me go ahead and erase that guy, and now we need to go look for some other cells on our list, and those are cells of Leydig. I'm going to make that go away, and then hopefully get back to somewhere near where I was. Yes. No, not quite. I liked where I was because the cells of Leydig in that area were quite good. I'm going to try and navigate back there. That's not quite where I was, but this is close enough. Also beneficial, you can see these little guys with the rice grain shaped nucleus. They look different from the other spermatids I show you. These are still spermatids, so this is a spermatid. And these are too. The first one I showed you here is not undergone spermiation yet. The second ones are undergoing it. They're changing form. So that's the difference there. Okay, cells of Leydig, aka interstitial cells. These are the ones that produce testosterone. So they're going to be positioned between tubules, but not all of the cells between tubules are cells of Leydig. So how do you differentiate? Because you can see these guys with the flat nucleus, those are going to be smooth muscle cells or fibrocytes. The ones that are close to the proximity, or the perimeter of the seminiferous tubules are going to be smooth muscle cells. There's a small amount of smooth muscle on the outside of every tubule, and then the rest of the interstitial stuff are mostly fibrocytes. So you have to be careful. Not every single cell that's outside of a seminiferous tubule is a interstitial cell. However, that's the only kind I'll point at. So these guys here that are darker, and that guy and these are cells of Leydig. So cells of Leydig make testosterone. And if you look closely, you'll notice that the cells of Leydig are positioned near not one, but two blood vessels. So if we know that hormones have to travel in the blood to reach their destination, and that's true of testosterone as well, it makes sense that there would be blood vessels nearby for that testosterone to enter and then go travel elsewhere in the body and do whatever it's supposed to do there. Okay, so let's zoom back out now, and I keep making that mistake, so silly. And let's go take a look at the epididymis. So before we zoom all the way in, I want to point something out, and that is that students tend to struggle with testis versus epididymis because if you can look closely, you'll notice both made of wadded up tubes that are cut up in a microscope slide, right? But if you look closely, you'll notice that the margin of the tube in the seminiferous tubule is very fuzzy, and it's irregular and disorganized looking, whereas the margin, or the apical surface of the tube in the epididymis, looks much more uniform, and you can see a clear difference between what's inside the tube and the wall of the tube. So this is a group of spermatozoa. This is the edge of the epithelium of the tubule of the epididymis. So let's zoom in and we'll do some comparing and contrasting. Okay, so this is classic epididymal tissue. Some major differences include still have smooth muscle and dense connective tissue surrounding, so that's not different. However, we have, instead of a germinal epithelium, a germinal epithelium is one which creates gametes. Instead of that, we have this pseudostratified columnar epithelium that is ciliated. So you can actually see all this cilia. And then you can see pseudostratified epithelium. So I'm not going to point at the wall of this tube and ask you what tissue type it's made of. We're past that stage in histology now. I assume that you can recognize different epithelial types when I ask you to. So the reason that I point these out in the videos is not because you should be trying to find the epithelium type and name it, but rather I'm telling you that's a distinguishing difference between this and the testis. So if you look at a tube and you suspect it might be testicular tissue, take a moment and look at the epithelial type and say, oh wait, no, that looks like pseudostratified ciliated columnar epithelium. We don't have that in the testis, we only have it in the epididymis, so that's what it must be. The reason I'm stressing that is because I see answers on practicals sometimes where students clearly missed the idea that I'm no longer going to directly ask you about tissue types unless the question explicitly asks for it. So I would put an arrow here, say, identify the tube that the pointer is in the lumen of. Or I would put the pointer here, for example, and a lot of times I'll get pseudostratified columnar epithelium because students saw something that was familiar to them and just named that instead of really reading the question. So being careful about what the question's asking and remembering that I assume now it's a foregone conclusion that you know your epithelia. The answer is never going to be lumen, and the answer is never going to be epithelium. That's for sure. unless I say tissue type or space for lumen, but that's unlikely. Okay, so you can see the cilia, you can see the pseudostratified epithelium, you can also see this big aggregation of spermatozoa in the middle. And notice that there is distance between the bolus of spermatozoa and the edge of the tube. That is pretty common in the epididymis as well, which is part of what gives the luminal edge of the epithelium its such a defined look. Okay, so that's that stuff. So now let's move on to another tissue entirely. So I'm going to leave testis and epididymis behind, and now we're going to look at the veins and arteries and the ductus deferens in the spermatic cord. So the spermatic cord is a group of tubes that take blood to and away from the testis and carry sperm away from the scrotum. So that's what we're looking for. So on your list, there's the ductus deferens, and then there's also the veins and arteries associated, as well as nerves. So this first ductus deferens slide, this one right here, is actually not ideal because I can't see very much of anything else. This one here is much better, so I'm going to click this one. Okay, so in paying attention to the labels of the slides is also important. So ampulla of the ductus deferens means that this is pretty close to where it joins up with the prostate. And so you can see, or excuse me, the seminal vesicle, and you can actually see the seminal vesicle over here. So over here is seminal vesicle. It's not on your list of things to know microscopically, so ignore histo 4. I'm specifying histo because I absolutely expect you to recognize the seminal vesicle on an image, but I don't expect you to memorize it histologically. Alrighty, so let's zoom in on the left side of the image and see what there is to see. Okay, so the lumen is in this center part, and you can see that it's convoluted, it's wrinkly. And then most of the epididymis is a thick tube of smooth muscle, and you can actually see along this dotted line I'm drawing the difference between the outer and inner layers of smooth muscle. So this is the outer longitudinal layer, and this one is the inner circular layer. So you can actually see the difference between the two of them. This is because sperm do not swim out of the male reproductive tract. They actually are propelled primarily by smooth muscle action and bulk fluid movement. So this smooth muscle is really important from that standpoint. Alrighty, moving right along. So the other thing on your list is the testicular artery, and then it says veins of pampiniform plexus. So let's go look at the artery first. Okay, here it is. So first thing first, how do I know it's an artery? Well, step one, there's red blood cells in the lumen. So all these little pink circles in here are red blood cells. And step two is I can see very clearly that the smooth muscle wall is thick. And the name for this is the muscularis. So if I see a thick muscularis relative to the diameter or size of the tube, and I see a lumen with red blood cells in it, and final step to confirm that you're seeing a blood vessel and not something else is to zoom in on the epithelium lining it. So I know that squamous epithelium is what lines blood vessels, so I'm going to go zoom in and check for that. But let me label this first. Looks good. So let me zoom in a little bit. And what you'll see is that the inner surface is lined with squamous epithelium, specifically simple squamous epithelium. So we've got a single layer of thin, flattened cells, which fits the criteria for it being a blood vessel. Now, the veins of the pampiniform plexus are a little trickier, and that's because there are more than one of them. The benefit to them being numerous is that you can find them pretty easily as long as you know what you're looking for. So let's find out what we're looking for. So for veins, same deal, blood vessels in the lumen, so this fits the bill. But you're going to see a collapsed lumen because the walls of smooth muscle aren't very thick and they don't hold the walls up very well. So see, this is all the same vein, but it's much more convoluted and irregular in shape because it has a thinner wall and therefore can't support a circular shape quite as well. So this is one vein. Let me go look for some others. Oopsie. Oh yeah, they're all over the place. So for example, here's some more down here. There's one. There's one. There's one. This is one. So you can see the veins are all over the place. So this is for a particular purpose, and that purpose is energy exchange and heat exchange. So the testes, in order to make sperm, they have to operate at about 1 degree lower than the male body temperature, which is about 37.0 degrees Celsius. So in order for optimal sperm production and to produce sperm that are viable and healthy, one must keep one's testes below that number. So part of the way that this is achieved is by moving the scrotum away from or towards the body. The other way that this is achieved is that if you run cold blood full of nothing but wastes and low oxygen past an artery that's carrying warm blood, heat is going to transfer from the artery into the veins. And the reason for that is easy to understand if you remember your thermodynamics and your diffusion laws from Biology 160, and that is the following. Just like matter, heat flows from an area of high concentration to an area of low concentration. So there's lots of heat in this guy, and it's going to radiate and end up in these veins. Which dissipates the heat enough that the blood reaching the testis via the artery is now the appropriate temperature for whatever the testes are at currently. So it helps to not cook the sperm as they're developing. These little guys over here are nerves, which are also part of the spermatic cord. Here's another vein. Vein. And then the rest of what you're seeing is connective tissue, empty spaces, and adipocytes. So that is the spermatic cord front to back. So finally, and I'm going to actually break this video up into 2 videos, one for XY individuals and one for XX. So we're going to go to the end of the XY section and we're going to look at the histology of the penis. So the penis is pretty easy to recognize because it looks like a little face. So it's got 2 eyes. And each of those eyes has a little pupil. And then it's got a mouth. So the 2 eyes, so to speak, are the corpora cavernosa. So corpora means bodies and cavernosa means cavernous. So the singular form of this is corpus cavernosum, And corpus cavernosum means a body with many caverns in it. So the cavernosum portion refers to all of these little blood sinusoids throughout the tissue that can fill with blood, and that's how the penis achieves an erection. Outside, here, there is more tunica albuginea. So this is that dense, irregular connective tissue that surrounds both the testis and also extends onto the penis to enclose these sinusoidal spaces and make them not stretchy. So when you fill something that's not especially stretchy with blood, what's going to happen is it's going to get very turgid, which means sort of erect, so that's the achievement of erection, or hard. So that's the prerequisite for intromission during sexual intercourse. If the tunica albuginea was absent, the sinusoids would still fill with blood, but if this tissue was elastic, the penis would never become rigid. So it's the tunica albuginea plus the blood inside of each of the corpora cavernosa that cause the ability to create and sustain an erection. Now, this bottom part I'll show you momentarily once I take well, actually, let me add something first. So this guy right here is the corpus spongiosum. Now, if you look carefully, you can see that there is dense connective tissue around it, but it's very, very thin compared with the tunica albuginea. That is because the corpus spongiosum is indeed an erectile tissue as well, so it does achieve erection along with the rest of the penis, but you don't want the pressure in there to get so high that it shuts this little mouth and blocks the passage of sperm. That would be no point, right? So it doesn't get quite as hard or rigid because it can't if it wants to perform the job of introducing gametes into another person. That's how it does its function. So this, of course, being the urethra, through which both semen and urine pass, but, quote, not at the same time, unquote. This is the traditional understanding of urination, that it is completely separate from ejaculation. But, you know, there are exceptions to a lot of things in human biology, and there is one to this as well. But in most cases, you should not have those two things occupying the same space at the same time. Why? Well, spermatozoa are really, really vulnerable to temperature changes, obviously, but also to acids, even relatively mild ones, such as those secreted by the bacterial flora in the vagina. So, sperm basically need to be kept away from the acidic stuff. So we'll get to more about how that works when we talk about the science around and behind ejaculation and fertilization, and then what makes sperm viable, what makes them non-viable, what does that mean, so on and so forth. But not right now. Right now we're just still learning structures. Okay, so I'm going to zoom in and show you a couple more things on the penis, and then we're going to finish up the male repro video, and I'll move on to the next one. Okay, so zooming in, you'll see some dense connective tissue forming the tunica albuginea. There are also some nerves here. Isn't that nice? Oh, I don't want to draw a line. Those are nerves. Here's a vein. So let's cruise down into the erectile tissue and see what there is to see. So right away, I'm noticing a lot of space in which blood could be, but currently isn't. I'm also noticing this guy right here, which is a deep artery. And there's one on the other side too. So during an erection, the deep artery permits blood to exit it when it otherwise wouldn't. And that blood fills up the spaces between these dense connective tissues, fills up that space. It's enclosed and has pressure put on it by the tunica albuginea, and that is what permits the achievement of erection, is basically putting a bunch of blood into a tube with spaces for it and then not letting it out. So more blood in than blood out equals penile erection. And I do want to stress that because all external genitalia are homologous, that means that the structures of the clitoris that perform erection, and there are many, also do this exact same thing, although not in the same shape. So erectile tissues in general look like this, where they have a deep artery and they have sinusoidal spaces that can either contain blood or not contain blood, depending on the physiological need of the organism. So let's see what there is to see down in urethra territory, and then I'll call this video good for a little bit. So same general scheme, except for instead of a deep artery, we have a urethra, which isn't to say there aren't arteries. There are. Here are a couple. Because blood has to get into those sinusoidal spaces from somewhere, right? It can't just suddenly spontaneously appear. The other notable feature of the corpus spongiosum penis is the urethra, which is large. It is lined with stratified epithelium. Remember, stratified epithelium was that weird one, or excuse me, transitional, my bad. Sorry, I need more coffee. Transitional epithelium, which is that weirdo one that is exclusive to the urinary tract. So you don't find transitional epithelium anywhere else. So let me label this for you. Urethra. And these are arteries. Not that you can read that. The final clue that you're looking at the corpus spongiosum and not one of the other ones, because I could zoom you in a little bit further than this, are these guys. So these are what are called urethral glands, and they help to produce secretions that lubricate the urethra and have antibactericidal sort of compounds in them to help keep the urethra clean as well. And this is important because the urethra is open to the outside world, but it's warm and it's moist and it's dark. And those are the three preconditions that bacteria just love, as well as yeast. So having an apparatus in the genitalia region where everything's kind of warm and damp all the time that is self-cleaning and self-sustaining is really important. So that's one of the reasons the urethra is there. Okay, folks, that's all for this video. This was the tour of the male reproductive system, and I will see you back here for the next video on the tour of the female reproductive system.